Lesson 15, the Endomembrane System. The endomembrane system is a group of interconnected organelles that work together to synthesize, modify, package, and transport proteins and lipids. This system is crucial for maintaining cellular organization and communication between organelles. The major components of the endomembrane system are the nuclear envelope, endoplasmic reticulum, rough and smooth ER, Golgi apparatus, lysosomes, vesicles, and the plasma membrane. These organelles are connected either directly, through continuous membranes, or indirectly by transporting materials in vesicles. The endoplasmic reticulum, ER, is a network of interconnected membranous sacs and tubules that plays a key role in modifying proteins and synthesizing lipids. These functions occur in distinct regions. The rough ER is responsible for protein modification, while the smooth ER is involved in lipid production. The hollow space within the ER tubules is called the lumen or cisternal space. Its membrane, composed of a phospholipid bilayer with embedded proteins, is continuous with the nuclear envelope. The rough endoplasmic reticulum, RER, gets its name from the ribosomes attached to its cytoplasmic surface, which creates a studded appearance under an electron microscope. Ribosomes on the rough endoplasmic reticulum, RER, transfer newly synthesized proteins into its lumen, where they undergo structural modifications like folding or acquiring side chains. These modified proteins become part of cellular membranes, including those of the endoplasmic reticulum and other organelles, or are secreted from the cell, such as protein hormones and enzymes. The RER also produces phospholipids for cellular membranes. Proteins and phospholipids that do not remain in the RER are transported via vesicles that bud from its membrane. Because the RER modifies and processes proteins for secretion, it is especially abundant in cells that produce large amounts of proteins, such as liver cells. The smooth endoplasmic reticulum, SER, is connected to the rough ER but lacks ribosomes on its surface. It plays a key role in synthesizing carbohydrates, lipids, and steroid hormones, detoxifying drugs and poisons, and storing calcium ions. In muscle cells, a specialized form of the SER, called the sarcoplasmic reticulum, stores calcium ions essential for triggering coordinated muscle contractions. Vesicles budding from the ER transport lipids and proteins, but before reaching their final destination, these molecules require sorting, packaging, and tagging. This process occurs in the Golgi apparatus, or Golgi body, a structure composed of flattened membranous sacs responsible for organizing and directing cellular materials. The Golgi apparatus has a cis face near the ER and a trans face on the opposite side. Transport vesicles from the endoplasmic reticulum fuse with the cis face, releasing their contents into the Golgi's lumen. As proteins and lipids move through the Golgi, they undergo modifications, such as the addition of short sugar chains. They are then tagged with phosphate groups or other molecules to ensure they reach their correct destinations. The Golgi apparatus packages modified proteins into secretory vesicles that bud from its trans face. Some of these vesicles release their contents into other parts of the cell, while others fuse with the plasma membrane to secrete substances outside the cell. Cells with high secretory activity, such as salivary glands or immune cells, contain many Golgi apparatuses. In plant cells, the Golgi also synthesizes polysaccharides, which are used in the cell wall or other cellular structures. Lysosomes, known for their role in digestion and recycling of organelles, are also part of the endomembrane system. They use hydrolytic enzymes to break down pathogens that enter the cell. In macrophages, a type of white blood cell, this process occurs through phagocytosis. The macrophage's plasma membrane engulfs a pathogen, forming a vesicle that fuses with a lysosome. The lysosome's enzymes then destroy the pathogen. The endomembrane system is a network of organelles that synthesize, modify, transport, and break down biomolecules. The rough ER produces proteins, while the smooth ER synthesizes lipids and detoxifies the cell. The Golgi apparatus further modifies proteins and packages them for transport. Lysosomes digest waste and old organelles, while vesicles transport materials. Finally, the plasma membrane receives vesicles for secretion or membrane integration. See you in the next lesson.